So I'm going to talk about the updated ribosomal RNA databases. These are sequences that you can use for identification and phylogenetic analysis of fungi, bacteria, and archaea. As I said a minute ago, Barbara Robertson is here, who's the fungal RefSeq curator, and I'm Peter Cooper, and I'm going to be doing the presentation, although I'm not a subject matter expert on this. So there's an outline of what we're going to talk about today. We'll talk a little bit about what targeted loci are, which is a name that we give to these kinds of sequences we're going to talk about today. A little brief background on the structure of ribosomal RNA genes. I'll talk a little bit about the scope and content of the NCBI RefSeq ribosomal RNA gene projects, which include the 16S bacterial archaeal, the fungal ITS, internal transcribed spacer region, which is the largest one, but we also have a large subunit and a small subunit database. We'll talk about searching and retrieving the data, and we'll do some live demonstrations. Okay, so we talk about things at NCBI called targeted loci. These are things that people think of as marker genes, barcode sequences, and a person that I work with calls them measuring tape sequences, which I think is very nice. You use them for phylogenetic analysis or for taxon identification. And this is molecular identification of taxa as opposed to traditional morphological means where you're keying something out or looking at the structure of a morphology of a colony on a plate or something like that. Some examples of these kinds of sequences are cytochrome oxidase barcode sequences, some protein coding genes, various ones are used, the mitochondrial D-loop sequences, and the ones we're going to talk about today, which are the structural RNA genes. For prokaryotes, there is a 16S, a 23S, and a 5S. For eukaryotes, there is an 18S, a 5.8S, and a 28 or 25S, depending upon the group of organisms. And there's a special kind of sequence that's more variable, that's between the 5.8S and those other two subunits. They're called the internal transcribed spacer regions, ITS1 and ITS2. And there's also a 5S RNA gene. We make sequences for these things. We call them reference targeted loci. We have these records that are available from RefSeq. Also, one of the points I want to make to you today is that there are BLAST databases that we've already made for you that you can use. The ribosomal RNA genes from, sort of microbes in the traditional sense of that word, in the old-fashioned sense, which includes the prokaryotes, archaea, and bacteria, as well as fungi. The real good thing about these sequences that we produce is that these are associated for the most part with taxonomic type material. Type material, of course, is the material or specimen or culture that the new species descriptions are based on. And so this ensures that the correct name is tied with the sequence, which has been a big problem with sort of sequences going into GenBank where the identity may be incorrect. So what are these useful for? These are useful for confirming organism identifications. If you have an unknown, they are good for assigning a preliminary ID, or at least a placement in a taxonomic group. You're not necessarily going to be able to identify the species, and we'll talk a little bit more about that. And you can use these to construct phylogenetic trees, or to put them in your phylogenetic trees to get some anchor points and things like that. Okay, so let's look a little bit at the structure of the ribosomal RNA genes in prokaryotes and eukaryotes. Talk about where these sequences are with reference to those two things. So here is an example. This is using our graphical sequence viewer to look at the ribosomal RNA cassette. There are multiple of these in a lot of bacteria. In fact, this is E. coli. There are 7 copies of this in the E. coli genome. There is a 16S gene that's shown on the left-hand side of this slide here. And that's the one that we have a corresponding record that we're going to be talking about today. So this is the RefSeq RNA for this 16S, and this is just showing a BLAST alignment to that part of the E. coli genome. Here's the 23S and the 5S ribosomal RNA genes there. These are the main reagents we're going to be talking about today for bacteria. And then these are the structural RNA genes for a fungus. This is a yeast, Kazakhstania africana. And you can see the structure of the RNA cassette there. This is the 5S RNA, the 18S RNA, the 5.8S RNA, and there's a 25S, the large subunit RNA. So this guy right here, the 5.8S, there's a space between the 18S and a space between 25S. This is the internal transcribed spacer regions. There are 2 of them, one on each side. These are more variable than the conserved RNA genes themselves. And we make a sequence. This is an internal transcribed spacer region RefSeq that includes that 5.8, but it also includes some of the flank on either side. So it includes that ITS1 and the ITS2 sequence. And so that's a very useful kind of sequence to use for identification and phylogenetic analysis. Okay, so a little bit more about these targeted loci data sets that we have. One way to access them is through the bioprojects site. There are a number of these. I've highlighted the ones we're going to talk about today. The 16S for Bacteria and Archaea, the fungi, the internal transcribed spacer region, the ITS, the 28S, and the 18S. And those are the bioproject IDs. These are all part of these targeted loci projects that we have at NCBI. What's good about these bioproject identifiers is they're very useful for retrieving these sequences, these sets of sequences from the Entrez system, and I'll show you that in a little bit. So looking at what kind of organisms or what kind of taxonomic distribution we have for the organisms in the groups. For the 16S, this is a breakdown of what groups are represented. And what I've done here is I've used sort of a name that's sort of a handle for a group of bacteria. These are the so-called BLAST names that we have in our taxonomy database. And so these are groups that most people recognize. The highest, most represented group are the high GC gram-positive bacteria. And then there's a number of other organisms represented here. So we have a broad spectrum of things. Of course, we'll talk later on. This is just a tiny bit of the microbial diversity that's out there. There are 21,333 of these records, 20,000 of them for bacteria, 979 for archaea, and it's about 16,000 species at this point. The fungal database is smaller. There are 11,751 records, 11,665 species represented. And again, this is a breakdown of the ITS fungal here. There are also the 28S and the 18S. They're slightly smaller databases. This is again breakdown by BLAST names. So these are not necessarily names that fungal taxonomists would like to use. Barbara didn't like them very much actually, but they're good handles for groups of fungi. And this is a little bit better breakdown of the taxonomic coverage, basically by showing you the number of records at various levels, kingdom, phylum, class, order, family, genus, and species for the small subunit, the ITS, and the large subunit. And Barbara made this slide. It's very nice, and it shows you the number in the various groups. So Ascomycetes, there's the largest group that has 8,492 records, and then so on down to the species level. Also, in terms of phylogenetic analysis, a lot of times people want to use combinations of genes. And the table at the bottom of this slide shows you what we have for a particular taxon. So for example, the last line are the ones that we have everything for, where we have the small subunit, the ITS, and the large subunit. And so that's 1,402 taxon that have that. And she has for you those that have the most taxa represented in these classes. And this is a breakdown by phylum, class, order, family, and genus here. And these are useful if you want to use, both or as many of the genes in a phylogenetic tree. So you can get those from our FTP site or from the RefSeq FTP site, and I'll show you that in just a minute. So if you want to look at one of these records, and this is the flat file of one of these records, just to show you the kind of information that's on here, and you can see these in our Entrez system. First of all, the definition line is controlled by the RefSeq curators, and that means that the organism information in this def line is going to be updated with the correct organism. What happens a lot of times with the GenBank records is that the definition line organism will be out of date, even though the taxonomy is updated in the organism field. And the important thing about the def line when we talk about the BLAST databases or the definition line is that that's the thing that shows up in the BLAST descriptions. And a lot of people are looking at the species name from there. So that's why it's useful to use these RefSeq records, because we're going to ensure that that species name is the best one we can come up with. Here's a link to the RefSeq Bio project. There's a link to the original GenBank or INSTC submitted a record that was used to make this record. And what's very nice here is that you can go back, you can go to our bio collections to look at the culture collection here. And you can link to the outside culture collection directly and see about how would you order this particular culture to verify this yourself if you wanted to, or to do work on this yourself. This is a 16S record. This is for Lactobacillus delbrueckii. And of course, it has this RefSeq accession number here. And here is the corresponding kind of record for one of the ITS sequences for a fungus. This is Trichophyton ruberum. And again, you have the curated def line, the link to the bio project, and the link to the outside culture collection where you could order this particular culture if you wanted to verify or do work on this yourself. Okay, so let's talk a little bit about how you can get access to these data. You can get them directly from our Entree nucleotide system, and it's quite easy to download these because they're not huge sets of sequences. You can use these queries right here, the bioproject number followed by the square bracketed field in the nucleotide system to get the sets directly, and then you can just use the send to menu on the upper right hand side to download these. If you want the sets in a handy file that you can download, you can go directly to our RefSeq FTP site, and the URL is given here. And there's a targeted loci directory that has access to the archaea, the bacteria, and the fungal sequences. The other thing I want to mention, and actually the title of this webinar, because one of the things that we want to kind of promote is that you can get these as BLAST databases, and we're going to see in a few minutes that they're also available on the web, of course, and that's what we're going to be using for our examples. They're right there on the FTP site, and the BLAST FTP site in the DB directory. You can get the ones we're going to talk about today, the 16S, 18S, 28S, and the ITS. These are the RefSeq databases. I will point out, and you'll see them if you go there, that there are sequences that we have gleaned from GenBank for these particular regions from other organisms. I'm not going to talk about those databases today, but you're welcome to check them out. One of the really cool things about the BLAST databases now is we have these version 5 BLAST databases so that the taxonomy information is baked into them. And so you can use some switches on the BLASTDB command program to dump out various things from the database, what organisms are in there, what the accession numbers are, and all those kinds of things. So that's very handy. The other thing that's nice about getting the BLAST databases, even if you don't run BLAST against them directly, is that you can use them as a way of dumping out the sequences in various formats. And this is a really nice way to get the FASTA that you could use in other applications. One of the things that I should point out is that the BLAST databases are non-redundant. And so there may be cases where the sequence for different species is the same. They're going to be collapsed into a single FASTA, although you will still be able to see the two titles in the BLAST database. So if you really want to have every FASTA sequence, you want to use the RefSeq download if you want all the records instead of the non-redundant set that's part of BLAST. So let me just point out a few things that are important to realize when you're using these kinds of targeted loci to do certain things. One of the important ones, and I sort of alluded to this earlier, is that this is limited coverage and taxonomic representation. So if you just do a quick count of the species level nodes at NCBI for bacteria and fungi, and again, these are often things that don't have formal names, they're just things that are at the end of the taxonomy classification. There's 450,000 bacteria, 150,000 fungi. For bacteria, we have only 16,000 sequences or species in that 16S database. For fungi, there's only 12,000 in the ITS database. Who knows how many species of microbes there are on the planet? I have seen estimates upwards of 10 to the 30th species. So this is just the tip of the iceberg in terms of microbial diversity. And so if you have an unknown sequence, there may not be the corresponding species in these databases. Also, just using 16S and ITS alone, there may be a lot more species out there because there's going to be some species that are not separable based on the ITS or the 16S alone, and you need to use a multi-gene analysis to get accurate identification and to get accurate phylogenetic trees. Now, The technique that we're going to be talking about today that searches these is, of course, BLAST. And if you go to the BLAST page now, you have a separate radio button that I should have highlighted here for you that you can click, and you have access to these 16S, 18S, 28S, and the ITS databases from the pull-down list. You can also restrict only those from type material, and most of these are from type material. So that's all very good. One thing to keep in mind, and we'll see some good examples of this in a few minutes, is that there are some limitations that BLAST has. One is that BLAST sorts by the E-value and alignment score and not by the percent identity. And this causes some things to happen that may be confusing at first. And that's that a longer sequence that's a worse match may appear higher in the results. So you need to take a look at both query coverage and percent identity to help identify things. Also, BLAST doesn't use position-specific information in the alignment. So every position in the alignment gets equal weight, but in reality, certain positions are more informative than others. So you need to look at how the match fits into a tree built from the results, for example, to get a better idea of how the placement of the sequence is. You also may need to build trees using real multiple sequence alignments and tree building using multiple genes to get an accurate view of things. Okay, so let's do some live examples. And so I'm going to switch over to a web browser, and this is where it would be useful if you wanted to follow along to try to get the text record, the text document that's on the FTP site. So let me get out of this. This is the one that I wanted. So if you go to the site, you can see this is the sort of run through that we're going to do for the demos. And the first thing I want to do is just do a simple BLAST search and a search to find a particular targeted locus sequence. So what I'm going to do is use one of those queries that I talked about where you're searching for the bio project, and I'm going to add an organism search to this. This is an Archaeon that we have a 16S sequence for. So I'm going to go over here to the nucleotide database. And notice that we have two different 16S ribosomal RNA sequences, and that's because there are these are divergent from each other. So we make two different sequences. And what I'm going to do is, this is an unusual kind of situation. This is an organism that has two central replicons, and we can actually find this cassette in the genome. So let's do that as an example, just to show you how these things line up, and take a look at an RNA operon in one of these organisms. So let me click over here to run BLAST. What that's going to do is to put my two sequences into the BLAST form here. I notice that we could search against the RNA ITS databases, but we're not going to do that right now. What we're going to do is to go ahead and search against the genomes. So I'm going to search against the representative genomes, which are a special set of genomes at NCBI, and this happens to be one of the ones that's in there. And I'll go ahead and type the species name in there to sort of limit our search. So I want Haloarcula maris mortui, which is the organism we searched with before. And we can go ahead and run that. Now, if you want to retrieve the results, if you didn't run the search yourself, you can do that pretty easily from the URL that I've given you here. So I've saved these BLAST results. So if you just copy and paste that URL into your web browser, you should be able to just retrieve them. I've got mine back here. And so notice that we have a hit on the two different chromosomes. There's a set of results for the first sequence. The first sequence matches as chromosome 1 perfectly. The first sequence matches chromosome 2 less perfectly, 94.92% identity. You can just see if you switch over to the other sequence here, that we've now swapped them, okay? So this is matching the other set. And if we want to look at this, we can jump down to the alignment here. Let's take a look at the graphics view, which is a nice way of looking at your results here. And there are the two sequences that we search with. And if we zoom out a little bit, we can see that, now this is just on the opposite strand here, but there's the 16S, the 23S, and the 5S, and here is our perfect match to this particular portion of chromosome 1. And so this is a case where we made two different 16S sequences because of the divergent 16S regions on this particular organism. Okay, and let's do another exercise with 16S. And what I have here, if you wanted to follow along, there are 3 different sequences from some bacillus species that don't have a species identification on them. So what we can do is to try to find the best species match for these against the 16S database. So I'm going to copy those. I can go back here and edit my search to get to the BLAST page if I want to, and I can reset the page just to start over again. And so this is our first look at the RNA ITS databases, ribosomal RNA ITS databases here. I'll select the radio button, and the top one, the first one, is the one we want, the 16S ribosomal RNA sequences. I won't change any of the other parameters. I can leave this, I can check for sequences from type material just to make sure that I'm looking at the type strains here, and run BLAST. And so there are 3 sets of results here now, the 3 different ones that we searched with. And let's take a look at this. So we have some potential matches here. One of the things I want you to notice, and this is the point that I made earlier, there are differences in the length of these sequences that we have made. And so what happens is the best match, which in this particular case is Bacillus strentensis, is lower down in my match. So what I want to do a lot of times to make sure that I'm getting the best percent identity is to go ahead and sort by percent identity. And then I can see that my best match is Bacillus strentensis. And that might be, and then there's another one here for a different strain of Bacillus strentensis. Another thing you can always do with these to sort of verify things is to look at the distance tree of results. And we'll do that more than once today, just to see how it fits into the tree. So you can expand this node here, and then you can zoom in to see what's going on with that. And so there it is, fitting right between those 2 Bacillus strentensis sequences. And so now, do I know that that's the species that it is? I don't know for sure. That's I've got it certainly within, it's close to that particular species. And I won't go through the other ones. It's pretty much exactly the same exercise. And the reason this is happening is because this NR here, this sequence here, is shorter than the other ones, and that's why we get a lower query coverage here, even though the percent identity is higher. So that's why those matches might be dropped down a little bit in your BLAST results. Okay, so let's go on and move on a little bit. So let's move over to a fungal clone here. I'm going to retrieve it just so you can see what it is. I'll go ahead and retrieve this from the nucleotide database. And so this is from what we call a pop set at NCBI, where people have submitted a whole bunch of sequences. In this particular case, it says it's an uncultured shoal fungus, and there's a clone, and we don't really know what it is. They have identified that it contains the 18S, part of the internal transcript spatial region 1, and so on and so forth. So we can run a search with this. And again, we can do the same thing we did before by running BLAST. And again, we're going to pick one of our RNA ITS database, but this time let's pick the ITS. So this is a fungal sequence. We'll go ahead and do a blast search with that. And so again, we have to make sure that we're looking at the best matches here. So we want to make sure that we sort by percent identity. And if I do that, then my best match turns out to be the Penicillium subrubescens here. Now, is this the species that we're sure that it is? We can't be sure, but this is the best match. And if we can, again, we can take a look at the tree if we want to see where that fits into the tree, see if that sort of confirms what we're thinking about that. And zoom. And it still comes out to be the closest thing. It's in this group of organisms here. So those ones that I showed you worked pretty well. And the last thing I want to do is a fairly complicated one. It involves a number of different steps, but I want to point out to you on the last one, basically that there's some things you need to think about. The database and the length of the query sequence and all those things matter in terms of the ID that you might get. So I'm going to go back. Okay, so I've got it over here. So this is a fungal sequence that Barbara provided to me. And let's start out by doing a search against a different database, one we haven't done a search against yet, which is the default nucleotide database. So I'll change my database to standard nucleotide databases here, and I'll leave my limit to type material on. Let's go ahead and run that. For the moment, let's just look at our best matches here. We have this Cautionary species. There's a bunch of Cautionariuses here. Cautionarius sesky is the one that looks like it's the best match. Let's take a look at that match for a second. I just want to point out something to you. So if we take a look at the graphics link, it'll give us some information about what this match looks like. But the other thing it points out is that the sequence that we're dealing with, our query sequence, incorporates more than just the ITS regions here. So here's a 28S, and so this thing has a big chunk of the 28S in it. So that's an important thing to keep in mind. So for example, we could think about doing a search against 28S, but they're more conserved in the regions down here that we're interested in. And so you can see that basically right here by looking at where the mismatches are. So we've got mismatches in the ITS regions, nothing really within the 5.8S, and nothing really within the 28S. But it does tell me that my sequence is longer than just the ITS region. Let's go back and run this against the ITS type strains database and see what that comes out like. So we'll change over to ITS here. And we'll run this search. So we now have a different species at the top. This is Cautionarius phoeniceus. And notice that you can see that the query coverage is 71%, points out something that we pointed out before, that our query sequence is longer than this ITS sequence that we're matching. And notice that Cautionarius sesky, which we had before, is now number 3. Now let's do the thing we did before. We'll take a look at the distance tree results and see what comes up when that happens. Expand that and zoom to look at it. So now When we look at the distance tree of results, the closest species is this one, Cautionary cinnamomous. And if you look up here, you can find the other ones in the tree. They're at different parts of the tree, the ones that we had matches before. So we now have a different closest match. Let's do one more thing, the thing that we've been doing all along, which is to sort by percent identity. And now we have Cautionary croceus as our best match. So that's the one that gives us the best percent identity. So we have we've identified 4 different closest matches, the Cautionary species Sesky, Phoenicius, Cinnamomous, and Croceus. Which one is it? That's a good question. And so in order to figure that out, you may find that you can't figure it out. And in fact, there's a slide that Barbara provided for me that shows you what the phylogenetic analysis was like when they did these species that we've looked at just now. And I'm going to switch back to the slides for a minute to show you the results of that. So these are the species that we've looked at, the Croceus, the Cinnamomous, and Phoenicius. So they're all in here. They're closely related to each other. And it shows you where these things are in the different things that we looked at. All we can say is that it belongs to this species group probably, but we can't give you a clear species identification for this. And that's just sort of a cautionary tale of the kinds of things that you're going to run into with using BLAST as a tool, and the fact that we have a limited taxonomic coverage for these organisms. Okay, so we have about 10 minutes or so for questions. And I'll put this slide shows you where you can get some more information. These are basically our resources for learning about things. If you have any questions, you can write to the help desk at that info address. And Rana, let's open the floor to see if there's any questions that we can answer.
Sure, there are actually a whole bunch of questions. So the first thing we'd like to talk about initially are the databases, the targeted loci database. There's a number of questions about what types of sequences are actually located in that particular database. One of the questions was about what kind of sequences are there. One of the people on the webinar was mentioning how there are many databases out there that have different levels of qualities of sequences. And what exactly goes into these targeted loci databases? What's the level of curation? Is it possible to maybe expand these databases, but then fragment them by those that have really good curation versus those that may be part of uncultured taxa or metagenomic profiling? What goes into these targeted loci databases?
So that sounds like a question that I think Barbara would be best at answering. Barbara, would you like to talk about that?
Yes, I can say a little bit about this. So for the ITS RefSec database, first of all, we do a lot of sequence quality control, looking at non-ATG characters so that there's not too many of those. We allow only like 0.5% characters to be in our data set per sequence. And we look at the assembly, making sure that the sequence is correctly assembled. We use the ITSX program to make sure that everything is present, that the complete 5.8S region is present. We also use that script to check how complete our sequences are. So some of our sequences will have a flank on the small subunit side and a flank on the large subunit side, but it will never be longer than 100 bases. So about, I would say, 50% of our data set has actually complete ITS1 to ITS2 sequences, and those where they are partial, we have generated a statistic where we look per genus. What is the expected length for the ITS1? What is the expected length for the ITS2? And if we cover 80% of either, we allow it to go in. Otherwise, if we're too strict in just, housing totally complete ITS 1 and 2 sequences, then we'll have very few coverage of our fungal kingdom. Let's see what else. Yeah, we look at make sure that there's no bookkeeping errors. We check the sequence name association, and we check, that sequences do not sit in weird places in the phylogenetic tree that doesn't make sense in terms of its classification in the NCBI taxonomy. And I'll just stop here.
And Barbara, I would say that the same sorts of, you talked specifically about the fungal sequences, but the bacterial sequences are also curated in a similar way. And then the second part of the question was about making data sets available for the uncultured, extracting those. I don't know that we have any plans to do that, and there are certainly other places where you can get access to those sequences.
Yes, we don't have any plans to do that, but people can go to the UNITE database, and they definitely do that. They do sequence quality control, they generate clusters of it, and try to put species hypothesis names on it.
Okay, thanks. Rana, close to time, but maybe you can pick out maybe one more question that would be probably
okay,
so one.
Right, there's a number of them that are actually very popular, but let's again go into the sequences, and they're looking for guidance on particularly fungal identification, but also, and maybe you could extend this to bacterial identification as well, is ITS for fungal identification specific enough? Are there other genes that might be good to include, or would enhance the ability to identify fungal sequences, particularly to the species level, on top of that? And then is it possible to have more than just one, let's just say ITS and maybe another gene, in order to do the identification and then the tree generation?
Yes, so there's some secondary barcodes. That's definitely important, especially when it comes to trichoderma and fusarium. And one of those protein coding barcodes is called elongation factor 1, and that can be used. Yeah, in terms of other tools, tree placement tools is probably the best tool to use. I know there's a group called TBAS. They maintain a reference tree, and you can use your ITS or your elongation factor sequence, and you can search against the tree, and it will placement of your sequence will be shown in a highlighted fashion in their tree to show, is this like a fusarium, is this like trichoderma or whatever. I think they only do Pazizomycotina, so the filamentous fungi. Other tools, yeah, would just be the clustering tools, but I would steer away from that. I would rather do like a tree placement tool. Other barcoding sequences that one can use is things RPB1 and RPB2, but I think elongation factor 1 is probably the best go-to protein coding gene after ITS. It depends on the clade or the genus you're working with. I suggest doing some homework, and if you're working on a specific genus, go and see what the literature say in addition to ITS, which is the applicable gene to use.
We are essentially out of time, so what I think I'd like to do is go ahead and end the webinar. And thanks, Rana and Barbara, for participating in this. Thanks, everybody.